I'm Tim O'Sullivan, Assistant Professor of Immunology at UCLA. The purpose of this seminar series is to increase the exposure of early career faculty in a time where we haven't achieved equity and representation at scientific conferences and seminars despite equally innovative discoveries. Professor of Immunology at UCLA, getting some feedback. And today we're excited to have Tiffany Reese from UT Southwestern. Tiffany received her PhD from UCSF with Rich Loxley and continued her work with Skip Burgeon at WashU as a Damon Runyon postdoctoral fellow before joining the faculty at UT Southwestern in 2015. Tiffany is currently a Pew Scholar in Biomedical Sciences and an Endowed Scholar in the Department of Immunology. Tiffany's work focuses on the understanding co-infection. And the reason for this is because we're all infected with multiple pathogens at any given time, but very little is understood about how those pathogens interact and how they influence the immune system. So Tiffany's work has been really instrumental in understanding how parasite infections can reactivate latent herpes virus infections. And she's also shown that chronic viral and parasitic infections can actually reduce the immune response to yellow fever vaccination. And so today, Tiffany is going to be talking about bridging the cellular biology of co-infection studies to investigate the regulation of chronic viral infection. We're really excited to have you here today, Tiffany. And in a second, we'll just let Tiffany take over. I've mentioned in chat, if you have any questions for Tiffany, go ahead and write that into chat, and then we'll address that at the end of the talk. So with that, I'll let Tiffany take it away. Thanks for being here, Tiffany.
Okay, thank you, Tim, for that kind introduction. And thank you to everybody who's tuning in. And also, thanks to Tim and the other people that have helped to organize this virtual seminar series. I think it's great to give more representation to early career scientists. Obviously, there was a kind of, very clear need that arose during the pandemic, since we don't get out and give as many talks and things. But I think it's also really important just in general to highlight early career people as much as we can. So I will just skip right to it. So in the Reese lab, we kind of have multiple different areas of interest, but they all center in chronic infections and using mouse models. So using mouse models and particularly herpes viruses, but other viral infections, we try to understand the cellular response to infection. We also study co-infection, and so we're interested in questions of, how do immune responses to individual pathogens like a herpes virus or an intestinal helminth, how are they different with individual infections versus combinations of infections? And how do these kind of chronic infections with, say, an intestinal helminth change the course of infection with a herpes virus? And then a third area that I'll just mention, but I actually don't have time to talk about today, is fetal immune programming. So taking some of these mouse models that we use in some of our other studies, like intestinal helminths, which do not transmit from the mother to the fetus, we use these models to infect mothers or to infect female mice prior to mating, and then understand how infections with these different chronic infections in mothers influence the development of the fetal immune response to viral challenges. So since most of what I'm going to talk about today is really centered on herpes viruses, I'll start there and introduce herpes viruses. So these are large envelope double-stranded DNA viruses, and there's 3 different kind of subfamilies of herpes viruses: the alpha, beta, and the gamma herpes viruses. Since today I'm going to talk about gamma herpes viruses in a mouse model that we use, the gamma herpes viruses in humans that that some people study or are interested in are Kaposi sarcoma-associated herpes virus and Epstein-Barr virus. But because many of the herpes viruses, and particularly the gamma herpes viruses, are very species restricted, meaning that the human viruses will only infect humans, we use a mouse model gamma herpes virus called murine gamma herpes virus 68 that is very similar to these, particularly to KSHV. And so like all herpes viruses, really a defining feature of a herpes virus is that these infections persist for the life of the host. So they're never cleared. And, in many cases, herpes viruses aren't really making you sick, but they're always there and your immune system is actively controlling those infections. So murine gamma herpes virus 68, like KSHV and EBV, if you infect an animal with the MHV68 virus, it will initially undergo acute replication. And so for approximately 2 weeks, you have an increase in virus titer shown here that can be detectable. And, for humans, maybe if you got mononucleosis when you were a teenager, then you were having an acute infection with EBV. But oftentimes, this initial acute phase infection may go unrecognized, or it may just make you feel like you have a cold or something. But after a couple of weeks, that acute infection will resolve, but the virus is not cleared. It enters the chronic phase of infection. And so during chronic infection with these viruses, there's little to no viral progeny produced, or at least it's below the limit of detection. However, in the case of herpes viruses, this chronic infection is actually a dynamic process because the virus is maintained in latency by the immune system, but it does have the ability to periodically reactivate from latency. And when the virus reactivates from latency, you will see a small increase in viral titer and viral gene expression, but the immune system then forces the virus back into latency. In the case of gamma herpes viruses like MHV68, these viruses establish latency in cells of the immune system, particularly B cells, macrophages, and dendritic cells. So in all of our in vitro studies, we mostly use macrophages, primarily bone marrow derived macrophages for viral infection. So the signals that induce herpes viruses to reactivate are varied, and there are multiple different signals. Including, immunosuppression will obviously induce these viruses to reactivate. HDAC inhibitors, IgG, NF-kappa B inhibitors will all induce virus reactivation. And the immune system is really critical for forcing the virus to go back into latency. So this kind of balancing act is occurring all the time. Some of the signals that we've become interested in that will also induce herpes virus to reactivate and are going to be the focus of today's talk are parasite co-infection and oxidative stress. So like I mentioned, I'm going to kind of highlight sort of two different areas of the lab, one being more cellular or molecular biology, and another being more systemic or organismal biology, I guess. And so the first story I'll tell you is about oxidative stress, and this was done by a former postdoctoral fellow in the lab, Dr. Lily Tao. And what she found is that reactive oxygen species can regulate cytoplasmic DNA sensing, and that in doing this, they regulate herpes virus infection. And then the second story that I'll tell you is done by a graduate student in the lab, Christina Zarek, and she's using herpes viruses as a tool to probe this co-infection biology. So starting off with oxidative stress and reactive oxygen species. So, a few years ago when we started on this, if you asked me, what does ROS do in the context of an immune response or, in response to a pathogen, the first thing that would come to mind is like, well, reactive oxygen species and oxidative stress are anti-pathogen, at least in the case of some bacteria. And that's because, I'm sure we all learned, in your intro immunology or microbiology class, that if a bacteria is an intracellular bacteria and it's taken up in a phagolysosome, that reactive oxygen species and oxidative stress can actually directly kill that bacteria. But that's really only one of the functions, obviously, of reactive oxygen species. And yes, it is true that in some cases, reactive oxygen species can be antipathogen and directly kill phagocytosed bacteria. But there's a lot of other infections, particularly with certain types of viruses, where it's not that clear cut. So in the case of some viral infections, reactive oxygen species or oxidative stress is actually propathogen. So these viruses like KSHV, so a gamma herpes virus, or HIV or influenza, they actually thrive in an oxidative environment. So clearly reactive oxygen species and oxidative stress play different roles in an immune response depending on the type of pathogen. And that's because actually reactive oxygen species do a lot of really essential things in the cell, and they're not always detrimental. So obviously, if you have high levels of oxidative stress and lots of reactive oxygen species, that leads to DNA damage and lipid oxidation and potentially even cell death. But reactive oxygen species are made as part of our normal metabolic processes all the time by mitochondria. They are also produced by oxidases in the cell. And in more moderate or low levels, they play an important function in redox signaling. So many proteins can be oxidized by reactive oxygen species, like hydrogen peroxide. And when they are oxidized, that changes their signaling ability. And so this is yet another way in which signaling pathways are modified post-translationally. And in fact, it's been described by many other groups that mitochondrial reactive oxygen species are essential for signaling in certain TLR, NLR, and RLR pathways. So certain pathogen-associated molecular patterns that are recognized by these PRRs will induce particular inflammatory cytokines. And in fact, reactive oxygen species like hydrogen peroxide are really essential for optimal signaling through these pathways and the induction of the inflammasome. So in this case, high levels of ROS may actually be promoting inflammation. But if you think about that in the context of our virus, like MHV68 or some other viruses, that still doesn't explain why these viruses would thrive in an oxidative environment, because if high levels of reactive oxygen species are actually increasing interferon beta, for instance, that should suppress virus replication. So clearly this isn't the whole story. So that's where we started. And so, first we confirmed really that reactive oxygen species were increasing murine gamma herpes virus 68 replication, specifically in bone marrow derived macrophages. Because as I mentioned, these are one of the cells that, or macrophages at least, are one of the cells in the body that the virus actually replicates in and establishes latency in. So we use bone marrow derived macrophages as an in vitro model. And so if you infect these macrophages shown in blue that are untreated and measure virus replication, you see robust replication. And then if you treat these cells prior to virus infection with increasing doses of menadione, which is a chemical that will induce the production of reactive oxygen species in the cell, you now see actually increased virus replication in the presence of menadione in a dose-dependent manner, indicating that reactive oxygen species at these is actually increasing virus replication. And not shown here, we confirmed that these levels of menadione treatment were not toxic to the cells in any way. So of course, the first question was, does this increase in virus replication require type 1 interferon? So again, we have bone marrow derived macrophages, either wild type cells or cells that are deficient in the type 1 interferon receptor. And we either treat with vehicle or menadione prior to viral infection. And you can see in wild type cells, there's a significant increase in virus replication in the presence of reactive oxygen species. But in the type 1 interferon knockout cells, type 1 interferon receptor knockout cells, there's now no increase in virus replication in the presence of high levels of reactive oxygen species, indicating that this increase in virus replication requires the type 1 interferon response. And in fact, if we measure the production of type 1 interferon transcripts by RT-PCR, you can see that virus infection by itself in the white bar increases, obviously, the production of type 1 interferon transcripts, but reactive oxygen species suppresses the production of type 1 interferon itself. So how is type 1 interferon made after DNA virus infection? I'm sure this is just a review for virtually everybody watching this, but a DNA virus like MHV68, when it enters the cytoplasm, it's going to stimulate the cGAS STING pathway. And that's because cGAS recognizes double-stranded DNA in the cytoplasm. And when cGAS becomes activated, it produces the second messenger cGAMP, which then activates STING. STING, which is an ER-resident protein, will translocate to the Golgi, recruit TBK1 and IRF3, leading to phosphorylation of both of these proteins, and IRF3 then goes to the nucleus and drives the initial production of type 1 interferon. And of course, once type 1 interferon is made, you get a feed-forward mechanism through the type 1 interferon receptor, leading to ISG expression and more type 1 interferons. So, is reactive oxygen species affecting specifically the cGAS STING pathway? So to start to answer that question, we decided to eliminate the virus completely and just look at what was happening with the cGAS STING pathway. And so in murine cells, you can stimulate STING directly with the agonist DMXAA. And of course, when you stimulate macrophages with DMXAA, you see a high level production of the type 1 interferon transcript just a couple hours after stimulation. However, if you start treating these cells with increasing doses of metadion in the presence of DMXAA, you can see that we now suppress the production of type 1 interferon in a dose-dependent manner, suggesting that directly reactive oxygen species are specifically inhibiting the cGAS STING pathway in some way, leading to reductions in type 1 interferon production. And of course, if we neutralize reactive oxygen species, we can rescue type 1 interferon. So in this case, we are stimulating again with DMXAA and treating with metadion to induce oxidative stress. But if we also treat with the antioxidant NAC, you can rescue type 1 interferon production. So this data then suggests that type 1 interferon is being inhibited by reactive oxygen species, and that specifically ROS is somehow inhibiting the cGAS STING pathway. But where in the pathway is reactive oxygen species inhibiting? So to initially address this question, we went back to virus infection, and we took cells, either macrophages from either wild type mice or STING deficient mice, treated them with metadion to induce reactive oxygen species production, and then infected with MHV68. And you can see that in the STING deficient cells, we now no longer see increased virus replication in cells with high levels of ROS, suggesting that the effects of reactive oxygen species actually require STING. And then if we look actually at the activation of the signaling pathway, here we're by Western blot probing for phosphorylation of TBK1 and IRF3, which are downstream of STING, and stimulating cells, the macrophages, with DMXAA, either in the absence or presence. Of this case, we're just directly treating cells with hydrogen peroxide. And you can see that, in cells that are only stimulated with DMXAA, you have phosphorylation of TBK1 and IRF3. However, if the cells are also treated with hydrogen peroxide, so they have elevated levels of ROS, you get a reduction in the phosphorylation of TBK1 and IRF3, suggesting that the pathway is being suppressed. So since both phosphorylation of TBK1 and IRF3 are suppressed, that maybe could suggest that the actual activation of STING itself is being inhibited by reactive oxygen species. So just to introduce this, I'm sure most of you know, but STING activation is actually a multi-step process. So STING exists as an apodimer in the ER. It will then, in the presence of cytoplasmic DNA, it will bind to the STING ligand, so like cGAMP, and then you have a ligand-bound dimer. But for STING to be efficiently activated, it actually has to polymerize. So you have to get oligomerization of multiple of these dimers, and then STING, obviously, it translocates from the ER to the Golgi. And so oligomerization of STING is actually an important step in this activation process in order to get efficient induction of type 1 interferon. So we wanted to look to see whether STING was being efficiently polymerized. So again, Here we have DMXAA stimulation in the presence of or absence of menadione or reactive oxygen species, and then of course NAC in order to rescue it. So under normal conditions, if you just stimulate with DMXAA, you get phosphorylation of TBK1 and IRF3. And down here, we're actually looking on a non-reducing gel so that we can see formation of STING polymers. And what you see is in the presence of DMXAA, you get this higher molecular weight band, so representing polymerized STING. However, if the cells have been treated with DMXAA and have high levels of reactive oxygen species, you now get a suppression of this polymerized form of STING. And if we treat cells that have high levels of reactive oxygen species with the antioxidant NAC, we can rescue this polymer formation. So together, this data suggests that reactive oxygen species and oxidative stress may be preventing the activation of STING by blocking this polymer formation. But to really show that, we need to know whether there is a particular residue of STING maybe that is being oxidized. So, jumping forward a few steps, obviously, we did mass spec and found that there is a particular residue of STING that becomes oxidized in the presence of reactive oxygen species. So down here, you see in the absence of reactive oxygen species, this particular cysteine, which in mouse is cysteine 147, exists in a reduced state, and there's really no oxidation of this particular cysteine. However, in the presence of reactive oxygen species, we see that you now get oxidation of cysteine 147. And so that's just quantitated here. With the oxidized cysteine being in the gray, and you can see that the presence of menadione increases the oxidation of specifically cysteine 147. And so cysteine 147 in mouse is analogous to the human cysteine 148, and of course this is actually a highly conserved cysteine across many different species in STING. And it's been shown actually by other groups that this particular cysteine is essential for STING activation. And it's been suggested that that's probably because this particular cysteine is actually required for the polymerization of STING. So our model then is that, under normal conditions where you have low levels of reactive oxygen species and STING exists as an apodimer in its reduced state, that STING can bind to STING ligand and oligomerize, and then efficiently induce high levels of type 1 interferon in the presence of cytoplasmic DNA. However, if there's elevated levels of reactive oxygen species in the cell, this leads to oxidation of this cysteine, 147 in mouse or 148 in human, and that when that particular cysteine becomes oxidized, we don't know yet if this particular, if oxidized dimer can still bind to STING ligand, but either way, you get a reduction in the polymerization of STING. And then when STING can't polymerize, you see less type 1 interferon. So we then wanted to go back to herpes viruses and see actually if this paradigm might hold true in animals. So when one of the systems that we use in the lab for tracking MHV68 virus replication, as well as reactivation from latency, is we have a luciferase tagged virus. And so if you infect animals with this luciferase tagged MHV68 virus, you can track virus replication by measuring the luciferase signal over time. And so what Lily did is she pre-treated animals with menadione by IP injection for a few days prior to infection with MHV68. And then after infection with MHV68, she tracked virus replication by measuring the luciferase signal over time. And what she sees is that by day 4, in the animals that have been treated with menadione and have high levels of reactive oxygen species, we also see higher levels of virus replication, suggesting that increased levels of reactive oxygen species in mice or oxidative stress in mice actually increases virus replication of MHV68. In addition, she measured whether the interferon response was changed in these animals. And so here she's measuring interferon beta transcripts. And over here, she's measuring the ISG CXCL10. And so in animals that are infected with MHV68 and untreated, you see that MHV68 obviously leads to increased transcripts of both interferon beta as well as CXCL10. However, if the animals were pretreated with menadione to induce oxidative stress, we see a significant suppression of the interferon beta transcripts as well as the CXCL10. Of course, this ends up being dependent on sting, suggesting that reactive oxygen species in animals both leads to increased virus replication as well as, and that's probably because it's suppressing the interferon response in the animals. We then did the opposite experiment. So rather than inducing oxidative stress in the animals, we tried to neutralize oxidative stress by treating animals with the antioxidant NAC in the drinking water prior to infection with MHV68. And we see the predicted result, which is that if you suppress the production of reactive oxygen species or oxidative stress in animals, we get a decrease in virus replication. So to summarize this part of the talk, we found that reactive oxygen species regulate DNA sensing actually by inhibiting sting, and that ROS reactive oxygen species inhibit activation and polymerization of sting, leading to reduced phosphorylation of TBK1 and IRF3. And that this redox regulation of sting leads to reduced interferon production and increased MHV68 replication. So to put this in the context of what I introduced earlier, this really suggests that reactive oxygen species and oxidative stress have both pro and anti-inflammatory functions. And of course, that's in some ways we've known that before, but our data shows that it's probably because of the specific pathways that are being regulated by reactive oxygen species and the specific molecules, and then of course the combination of that with the specific pathogens. So certain pathogens and certain pathogen-associated molecular patterns, the inflammatory response is actually going to be enhanced by the production of reactive oxygen species. But for other viruses, perhaps, or in the case of just stimulation of the C-gas sting pathway, reactive oxygen species is actually leading to a reduction in interferon production by directly regulating sting activation. So obviously there's lots of future questions. What are possible endogenous sources of reactive oxygen species in the context of herpes virus infection? Herpes viruses have been shown to actually induce the production of reactive oxygen species. We focused in this story primarily on acute replication, but what's the effect of ROS really on latent and chronic herpes virus infection? Work by others has shown that reactive oxygen species and oxidative stress induce herpes virus reactivation from latency, but is it by this particular mechanism? How is ROS modifying other pathogen recognition pathways in the context of stimulation of those pathways directly, or the context of the infection with those pathogens and the stimulation of multiple different pathways all at once? Do particular disease states associated with elevated oxidative stress have impaired interferon production? And could oxidative regulation of sting be a way to dampen interferon production in the context of autoimmune disease? So these are some of the questions that we still have related to this project. But now I'm going to shift gears and talk actually about my graduate students project, which is going to go more into the co-infection and what the lab studies related to co-infection biology. So Christina Zarek did all of this work, and it's really a continuation of, in some ways, what I've shown previously, which is that parasite infection or parasite co-infection can induce herpes virus reactivation. And, a reason to ask these questions or to be interested in this is that actually all of us are infected with at least one herpes virus. So herpes viruses are part of our virome. Here is a graph that's just estimating the burden of chronic viral infections in humans. And in fact, many of these viruses that are shown indicated by the red arrows are herpes viruses. So if every one of us is infected with 8 to 12 chronic viral infections, and at least one of those is a herpes virus, then yeah, they're really part of our virome. They're there all the time, and they're modulating potentially our immune responses to other pathogens. And the course of their infection is probably being altered by other infections or maybe vaccines or other stimuli. And this is important to understand because herpes viruses can be both harmful and beneficial to the host, and probably neutral as well. But there are contexts where they are harmful. They can lead to cancers in particularly people that are immunocompromised or have had maybe more reactivation events, but there's also contexts where they are beneficial to the host. And so maybe being chronically infected with a herpes virus actually can protect you against other maybe lethal bacteria challenges. So I think understanding how these viruses are regulated by our immune systems and by our immune responses to other pathogens is sort of important for understanding how they're affecting our basic biology. So way back now, it feels like eons ago when I was still a postdoc, I asked the question of, how does inflammation change herpes virus latency? And can we induce virus reactivation from latency through co-infection? And kind of borrowing on my experience from graduate school in looking at intestinal helminth infections, we chose to infect latently infected animals with the parasite Heligmosmoides polygyrus. So this is a mouse helminth parasite, and so it's endemic to wild mice. It's transmitted by the fecal oral route. It establishes infection in the intestines of animals, and it resides only in the intestines. And in certain strains of mice, including C57 black 6 mice, it does actually establish a chronic infection. So this fecal oral transmission, basically animals will ingest these L3 larvae. The larvae enter the intestines, they mature into an adult, they mate, they lay eggs, the eggs come out in the feces. Those eggs mature into different stages of larvae, eventually become L3s, and that's how the life cycle progresses. And of course, many people in the world are infected by soil transmitted helminths. A quarter of the world's population is probably infected by helminths. So it's a, I guess, biologically relevant question. So to answer this question, we decided to again use our luciferase tagged virus. And so this virus expresses Firefly luciferase under control of a lytic viral promoter. And so we used it prior to this, as I showed you, in measuring lytic virus replication. But in this context, we're going to use it to measure virus reactivation from latency. And that's because when the virus is actually latent, you should not have expression of the Firefly luciferase, but if the virus enters lytic replication or reactivates, then you should get expression of Firefly luciferase. So we took mice and we latently infected them with MHV68, waited for a month or more, and then challenged those mice with the parasite Heligmosmoides polygyrus. We then tracked virus reactivation in these animals and quantitated it here. And what we see is that after helminth infection, the virus reactivates from latency. And so then, of course, we were interested in knowing what was the mechanism, like why was the virus reactivating from latency? And of course, parasites are masters of immunomodulation. The reason that they can, these large, in some cases large organisms, right, can persist in a host for a long period of time is because they have figured out ways to modulate the immune system. And it's been shown by work by others that they also change the immune response to lots of other pathogens. So for instance, HIV, TB, malaria, and co-infected humans. And they're capable of modulating a lot of different pathologies in mice. anything from food allergies to inflammation to bacterial pathogens and vaccine responses. And these types of parasites, these helminth intestinal parasites, induce a strong Th2 type or T regulatory type immune response. So in the most simplistic view of things, a virus induces a strong Th1 response, but a parasite induces a strong Th2 response. And so when an animal is infected or a human is infected initially with a parasite like this, they produce large amounts of IL-4 and IL-13 and probably IL-5 as well. And so, as I'm sure all of you know, IL-4 and IL-13 actually share the IL-4 receptor. They both signal through the IL-4 receptor, and they both induce phosphorylation of STAT6. And then STAT6 drives a Th2 immune response. But we first asked whether the virus reactivation that we were seeing in the context of parasite co-infection actually required this IL-4 STAT6 signaling. So we took animals that were deficient in STAT6, shown here in the green, and tried to co-infect those animals with both virus and then the H. polygyrus parasite. And what you see is that the parasite can no longer induce herpes virus reactivation from latency, suggesting that reactivation from latency in the context of parasite infection actually requires IL-4 and IL-13 signaling. Something about the immune response, right, to the parasite. And we could model that in vitro, again, taking bone marrow derived macrophages, infecting them with the virus in the presence or absence of cytokines. And what you see is that IL-4 as well as IL-13. Here we're looking at the staining with MHV68 virus itself, and you can see that IL-4 and 13 increase virus replication actually in bone marrow derived macrophages, whereas the cytokine IL-5 does not, and IL-5 does not signal through STAT6. And so I'm skipping a lot of data. Essentially, we came to a mechanism where we showed that actually the virus has promoters that are responsive to IL-4 and STAT6 signaling. So the virus encodes a promoter called, and it drives a gene called OR50 or RTA, and STAT6 actually binds directly to this viral promoter. And when OR50 and RTA are produced, they drive lytic virus replication and lead to virus reactivation from latency. So this was a very, I think, cool and neat mechanism showing that the virus has somehow evolved to sense a particular immune signal, in this case, IL-4 and 13, that may be generated by co-infection and sort of hijack that signal in order to induce virus reactivation from latency. It turns out that there's lots of other immune signals, though, that will inhibit that particular viral promoter. So this may be one way in which this balance between latency and lytic virus reactivation is maintained in a normal host. But, as you can imagine, this was a very specific setup for an experiment, right? Like the way we discovered this is we took mice, we latently infected them with virus, we waited 4 to 6 weeks, and then we challenged with helminth. And so that's really just testing, does helminth infection have an effect on virus reactivation from latency, specifically only in latency, right? So what happens when you start to mess with the timing? And so that was really the question that drove Christina's project. So does the order or the timing of this co-infection matter? And so this was the original setup that I just described. We had MHV68. We waited for more than a month and then challenged with parasite, and then we looked at latency and reactivation. But Christina decided to flip it around and ask the question of, well, okay, if you first have helminth infection and then you challenge with virus, what happens to virus acute replication, and then also what happens during latency? So first, let's look at latency. And actually what she sees is that if she takes either in blue are going to be the virus only infected mice, and in red are the virus and H. polygyrus infected mice. So these mice received H. polygyrus. 7 days later, they were then challenged with MHV68. We then waited a full month at least and looked at reactivation from latency. And so this is a slightly different assay than what I showed you previously. And in this assay, we're measuring how much virus will reactivate out of latency when we just take the cells out of the animal. And so what you see is that about 1 in 10,000 cells in the peritoneum, so we take cells out of the peritoneum, so 1 in 10,000 cells will reactivate the virus if they are only infected with MHV68. However, if they were pre-infected with parasite and then challenged with MHV68, you now see that about 1 in 1,000 cells will reactivate from latency. And this data really suggests that because we're not measuring in vivo kind of real time, I guess, reactivation, we're actually measuring the propensity of cells to reactivate out of latency when you remove them from the animals, that the co-infected animals have an increased propensity to reactivate. And what was really interesting is that this reactivation actually did not depend on STAT6. So somehow the mechanism of this increased reactivation when the timing of the co-infection is altered is now not dependent on STAT6. So here she's taking either wild type animals or STAT6 knockout animals and doing the same experiment, either single infection with only MHV68 or pre-infect with helminth and then challenge with MHV68. And if you compare just the black and the purple, which are the STAT6 knockouts, you can see that, the black looks very similar to wild type, so that's the control, and that in both wild type and STAT6 knockout animals, you get an increase in virus reactivation when the animals are pre-infected with helminth. So something is different about this mechanism now. If she then goes earlier and looks at what happens during acute virus replication, again, she sees that mice that have both infections, so they were pre-infected with parasite and then infected with MHV68, there's like a transient increase in virus replication at day 2, but that actually mostly normalizes, and in the end, the virus basically equivalently replicates or resolves acute replication equivalently in both animals. And the difference in this day 2 acute virus replication is also not dependent on STAT6. So again, whatever is happening between the parasite and the virus in this particular context now does not depend on IL-4 and IL-13 signaling. It's by some other mechanism. So to get at this, Christina started looking at what was happening to the cell populations in the peritoneum during either single or co-infections. And that's because the peritoneum, and specifically the macrophages in the peritoneum, which are one of the primary cells that the virus is replicating in, are significantly altered by both parasite infection and virus infection. And turns out that in the peritoneum, there are at least 2 different populations of macrophages under steady state. There's large peritoneal macrophages and small peritoneal macrophages. And these large peritoneal macrophages are kind of thought to be the more resident macrophage, but they actually derive from a yolk sac progenitor. So they're fetal derived, and they have a local self-renewing ability. On the other hand, small peritoneal macrophages are generated from a hematopoietic progenitor that comes from the bone marrow, and they derive from inflammatory blood monocytes that get recruited during inflammation. So, work by many others actually has shown that parasite infection, and particularly intestinal helminth parasite infection, can really alter these macrophage populations. So we started to think about that. And so under homeostasis, what you have primarily in the peritoneum is a large population of large peritoneal macrophages with just a few of these SPMs, or small peritoneal macrophages. However, if in the context of inflammation or infection with, say, a virus like MHV68, what normally would happen is the LPMs would reduce. This is called the macrophage disappearance reaction. It's thought that maybe a lot of these cells actually go to the omentum. And inflammatory monocytes would be recruited into the peritoneum, which would then become macrophages and become more small peritoneal macrophages. Perhaps some of them also end up becoming a large peritoneal macrophage subset later on. But what you would normally see is a shift from LPMs to SPMs if mice are infected with, say, virus. And as I mentioned, latency really occurs in B cells and a lot in macrophages in the peritoneum. So that's a major reason that we were interested in this. And the parasites, though, in contrast to, they don't really induce a lot of these SPM expansion. They actually enhance the retention maybe of LPMs or even the expansion of LPMs. And so we've seen that MHV68 infects macrophages in the peritoneum, but no one's really looked until very recently. It's like, which subsets of macrophages does MHV68 preferentially infect? And do the dynamics of the LPM and SPM population during co-infection actually change how MHV68 can establish latent infection? So if you look in the peritoneum at virus-only infected mice, what you see is what you would expect, which is that the large peritoneal macrophage population is somewhat reduced from what it would be in an uninfected state, and you get this significant expansion of the SPM, the small peritoneal macrophage population. However, if the mice are co-infected with both parasite and virus, now you see a significant amount of cells that sit and look like large peritoneal macrophages and much fewer cells that look like small peritoneal macrophages, suggesting that co-infection with the parasite is changing the LPM-SPM ratio. And so if we quantitate that, you can see total numbers of LPMs are significantly expanded in the co-infected animals. So that's the co-infected animals are red, whereas the MHV68 only infected animals are blue. And just for reference, black is HP only. So, so you can see that HP infection is leading to a retention of the large peritoneal macrophage population, and it's actually reducing the number of small peritoneal macrophages, whereas in mice that only have MHV68 infection, you get a significant increase in the SPM populations over time. But which cells are actually infected with virus? So we have access to another marker virus that was made in another lab that expresses beta-lactamase. And so we can use this virus to actually track the cells that are directly infected with a virus. And what you can see is that in this is just the total peritoneum and looking at CD11b positive cells. And here on the x-axis is staining for MHV68, so presence of the virus. You can see about 8% of the cells in a virus-only infected sample, most of them are macrophages, are infected with the virus. However, if the animals are now co-infected with both virus and parasite, there's a substantial expansion of this co-infected in these co-infected animals of the virus-positive cells. So about a 5-fold expansion. And if we actually quantitate that, again, you see that co-infected animals have significantly more MHV68 positive cells. And that's actually striking because, when we measured by viral titer, we only saw a transient increase in virus replication as measured by conventional plaque assay, and that was specifically at day 2, which is this time point. But we didn't really see any differences in virus replication at day 4 and day 7. However, we are seeing that there are actually more cells infected. So maybe some of these infected cells are actually latently infected and they're not acutely replicating the virus. And then if we subdivide these macrophages and look at LPMs versus SPMs, we see that the co-infected animals have significantly more infected large peritoneal macrophages. So it seems though the virus actually may preferentially replicate in large peritoneal macrophages and may, particularly if you look at these later time points when we didn't see a difference in viral titer, may actually be preferentially establishing latency in large peritoneal macrophages. Because if you look in the single infected mice, so the ones that only have MHV68, you see that in small peritoneal macrophages, normally the virus is replicating in the small peritoneal macrophages. So something about co-infection is actually maybe changing the reservoir of cells that are becoming infected with the virus. So I guess the take-home message I would leave you with now, because we have a lot of other experiments ongoing and a lot of stuff that's maybe not quite ready to show yet, but the timing of co-infection really matters potentially, both for the phenotype as well as for the mechanism. So if you have a latent herpes virus infection first, and then you come in with, in this case, an intestinal helminth, you can increase virus reactivation from latency, but this reactivation increase is transient. So I didn't really touch on that earlier, but the reactivation goes up and then it resolves and goes away. So it's a very transient event. And it depends on IL-4 and 13 and by that promoter mechanism that I described earlier. However, if you reverse the order of infection, you start to see something different. So if you first infect with parasite and then infect with virus, you see not only that there's an increase in virus reactivation from latency, but you see that there's an increased propensity of the virus to reactivate. So anytime we take those cells out of the animals, the co-infected mice will have increased reactivation, whereas the single infected mice will not. And that this reactivation does not depend on STAT6. What we think is happening is that parasite co-infection is altering the early infection events for MHV68 and specifically altering the resident macrophage population, which is then altering the reservoir for virus replication. Now, some of the things that we're doing that are ongoing is because this large peritoneal macrophage population has specific transcription factors like GATA6, which are important for their generation. We're doing experiments in GATA6 lysine cree mice, and we're also testing whether retinoic acid plays a role in this mechanism. And that's because retinoic acid has been shown to be important for the maintenance or the generation of these large peritoneal macrophages. It also turns out that retinoic acid is important in virus replication and reactivation from latency. So that may be another interesting tie-in. So I'm going to leave it there so that hopefully there's a little bit of time for questions. But first, I would like to thank the entire Reese lab. As I mentioned, Lily Tao did the first story and Christina Zarek did the second story. There's now multiple other people in the lab that have contributed in various different ways to all these experiments. We've got multiple collaborators that have been extremely helpful, and of course, our funding.
Thank you so much, Tiffany. So we usually have like a kind of a lag before the chat catches up with the end of your talk. So I'll just go ahead and get you started with the question while we're waiting for that. So I was kind of interested in the first part of the seminar, and I was wondering whether this inhibition of sting by intercellular ROS, like if you feel like that's some sort of metabolic adaptation as like inducing a negative feedback loop as a consequence of activation. Do you think that's what's going on? And like if you can look at that kinetically within like activation?
Yeah, I think that's a really interesting idea. And yeah, I mean, it basically plays into this idea that, pathways like this, like the STING pathway, is just, it's a highly regulated pathway. And there's probably lots of different ways post-translationally that STING gets regulated. It's regulated by trafficking, other post-translational modifications, oxidation. And I would think that this must be an adaptation in order to ensure that you can shut it off, maybe. So, too much STING activation is obviously detrimental in the context of autoimmune disease, but in infection as well. So there have to be ways to turn it off. And so maybe there is, like a metabolic feedback loop that contributes to this. I mean, one thing that I am interested in is like, well, if herpes viruses lead to some like relatively low level of oxidative stress in the cells just by infection during latency, like is one reason, right? It's probably many reasons, but is one reason that they do that because it dampens interferon signaling. And the virus, obviously interferon is antiviral. And so by leading to some oxidative stress, it may allow the virus to reactivate a little bit by suppressing interferon. But
Thank you. So we have a question in the chat that says cells are really good at modulating redox stress through oxidative PPP. Is there evidence that viral infections can modulate oxidative PPP, or does vitamin K malnutrition lead to viral susceptibility?
Sorry, vitamin K, is that what you said?
Yeah.
And malnutrition. Yeah. I don't know actually about, say, malnutrition or anything like that, but it is clear that these viruses like MHV68 do regulate, I guess, as I mentioned in the previous question, do induce oxidative stress, probably also have antioxidant mechanisms as well, just to regulate the oxidative balance in the cells. But I don't know if there is any data on like malnutrition or vitamin K as far as MHV68 goes.
Okay, and Joe has two questions, or maybe like a two-parter. And he's saying, in the absence of helminth, can a strong Th2 response from an allergen or cytokine, so IL-4, IL-13, be sufficient to drive the MHV reactivation from latency? And,
yeah, so, oh, sorry.
I'll get to the second part. You just ask that.
So we primarily did intestinal parasite infection, but we did test, we tested a schisto egg model. So the schisto egg model is not actually an infection model. It's just you prime mice with the schistosomiasis eggs, IV, and then you challenge again with schisto eggs. So basically you induce like a large IL-4/13 response, granulomas, all the stuff that happens in the lung, because basically the eggs end up lodging in the lung and induce this like Th2 inflammatory reaction. And we see a similar effect, which is that in the schisto egg model, you can also induce herpes virus reactivation from latency. So again, that suggests that that's not really an allergic model per se, but it's also not infection. And then the other thing that we did is we tried just injecting animals with IL-4 or IL-4 complexes, so longer-lived IL-4. And if you only inject with IL-4, you do not see herpes virus reactivation. However, if you inject with IL-4, but you also neutralize interferon gamma, then you see really robust, actually, even better than parasite by itself, even better than parasite. So if you inject with IL-4 and anti-interferon gamma, you see really robust virus reactivation. And so what we think that means is that, IL-4 and probably IL-13 is leading to like a positive signal, which is inducing OR50, so this viral transcript expression. However, you also have to basically neutralize like a negative signal. So, all the time that animals have some level of interferon in a latently infected animal. And so in order to get virus reactivation, you have to neutralize this latency signal or this negative reactivation signal and also give a positive signal. So the hypothesis would be that parasite infection, is able to basically do both somehow. But I guess we haven't shown that directly for the parasite.
So the follow-up to that from Joe is there clinical evidence that people with allergies or asthma have greater herpes virus reactivation?
I don't think anyone has looked. So if people, there have been some studies in Africa where people have looked at whether intestinal parasite infection correlates with increased seroprevalence to, say, KSHV, and there's a couple studies that suggest that there's increased seroprevalence. Now, whether that's reactivation or not is somewhat debatable, but that's suggestive. But I think, most of the time, gamma herpes virus reactivations are subclinical. Like they don't make you visibly sick. Like it's not like a herpes simplex virus reactivation, which might give you a cold sore, right? That's an alpha herpes virus. But yeah, an EBV or KSHV reactivation probably kind of flies beneath the radar most of the time. And I don't think anybody has directly looked at whether people with allergy and asthma have more reactivations or not.
Okay, I have one more question in chat from Isabel, and she's wondering if the latent MHV68 load differs in your H. polyinfected mice. She can imagine the reactivation could result in elevated or decreased load due to a kick and kill situation.
Yeah, that's a good question. So no, it does not. So, but gamma herpes viruses and particularly MHV68 is maybe somewhat unique in the sense that the level of latency is very hard to change. So there's the virus establishes latency at a very specific amount. And so in a normal animal, it's about 1 in 10,000 cells may be latently infected, and that doesn't necessarily change that much. And what we find in the co-infected animals is that the level of latency is pretty close. It's just really the ability to reactivate from latency. And so we think that maybe suggests that the virus can enter latency in both circumstances, but that perhaps the co-infected animals are not as tightly latent. So, latency may be maintained by a lot of different mechanisms, including silencing. So the MHV68 genome exists as an episome in the nucleus. And so it gets chromatin modified and silenced in the nucleus. And so perhaps co-infection is altering the ability of that genome to be silenced. And then it may also just be altering the cell type, and maybe certain macrophages, either LPMs or SPMs, have a different ability to actually reactivate the virus. But yeah, we don't see a difference in infection from latency.
Thank you. Yeah, well, it sounds like there's a lot of interesting open questions for your lab to explore going forward. So I'd like to thank our speakers today for joining us. Thanks, Tiffany. Thanks for everyone for joining us live. And if you didn't catch it live, it's still able to view because this is being recorded on our YouTube channel. So thank you everyone again, and we'll see you next time.